So we'll wait a couple more minutes and then we'll start. Good afternoon, everyone. Welcome to our session for the introduction of the metagenomic data analysis. My name is Farhana Musarrat. I'm a postdoctoral researcher at LSU and also the program coordinator for this course. So today we are going to give you a brief overview of the course, and then we will have two Q&A answer session, question and answer session. One will be on the March 4th and the other one will be on March 28th. So in this course overview, we're going to show you the course materials, and if you have any other questions, we'll try to answer those. So before we go to the course overview, let me remind you the registration process one more time. So most of the people already who are enrolled in this course, they have already registered. But if there are someone new and you are looking for the registration process, I'm going to go through them over again. So if you are a part of LBRN program, or if you are someone working at LSU, your registration process would be a little bit different than NAPI or any other who is not from LSU or Louisiana LBRN program. So if you are part of the LSU or LBRN program, you have to register through the iLab. Let me show you how you are going to do the registration if you are part of LSU or LBRN. So first, you have to create an account in the iLab. Once you have an account, you will be able to log in, and then these are the steps that shows you how to register for the course through iLab. So once you register, once you log in the iLab, you will be able to see the core facilities on the left-hand side corner. You select the core facilities, and then you select the CEDAR core facilities and services. Afterwards, you have to request a service, and it should be here on the top right corner. And then you will be able to select the courses that you are interested in. So in this semester, we are offering 3 courses or 3 training modules. One is the Introduction to Bioinformatics, which we already started. The second one is the genomic or metagenomic data analysis, which we already started, and the metagenomic is starting today. And the other one will be data science for biomedical data intermediate. And that is the other one we also started already. So you can take all three courses, or you can take whatever you want and select the courses here, write it down. And afterwards, this tab will come, this page will come. You don't have to write the fund number or anything, just write LSU or LBRN if you are part of LSU and LBRN, and then submit the request to core. And this is how your registration will be completed. And afterwards, we will take care of it. But if you are not part of LSU or LBRN, you don't have to do these, or you don't have to go through these procedures. I get a lot of questions, people who are not part of LSU, they are filling out forms, filling out this form and trying to register, but you just have to email me that you are not part of the LSU, but still want to register for this course, and I will direct you to the other link that someone will help you to register. This course is absolutely free for LSU, LBRN, or if you are part of the NAPI program, so you don't have to pay for the courses. So now I will show you, after your registration is done, what you are supposed to do. So once your registration is complete, you can create an account in the OmicsLogic. This is free. You can create an account. There is no charge for that. So once you create an account here in the OmicsLogic, learn-omicslogic.com, you can click on your email address here. This is your profile. So if you click on the profile, you can give us some information about you. Right? For example, you can upload your picture. It doesn't have to be your own photo. It can be your university logo, or it can be any other photos that you are comfortable with. You can upload some information here in the bio section, like what you are doing, what you are interested in, your names and your social media links if you are comfortable with it. So this will give us some information about you, and then you will update this information here. So in this page, you will see the courses, programs, and everything in activity, everything here. So if you click on the courses, you will be able to see the courses that you are registered for. And if you go to your profile and click the courses here, you will be able to see your activities and what are the courses that you took and how much you progressed, like percentage and everything. And also, if you click on the project, it will show you like how much you are done or completed in the profile, and what are the courses you took, and what are the how much points you got. So you can see here earn 10,000 points. That means if you earn 10,000 points, you will be able to do your own project using the pipelines provided by us. So every time you finished or you complete one course, you will get, or you will earn some points. And if you complete the 3 courses that we are offering this semester, it will be 10,000 points. So afterwards, you will be able to run your own project. So once you complete any project, once you complete any courses, you will get a certificate. And in the activity section here, it will show you how much progress you have done. For example, it's I have not done a lot of activities recently, so it's kind of straight line. But when you study and complete quizzes and stuff, there will be a lot of activity. You can see a lot of peaks. Okay, I think I have covered whatever I was supposed to. So this is the introduction course for introduction session for the metagenomic data analysis. And we will have 2 Q&A sessions in March 4 and March 28. You will receive an invite. Also, I will send you an reminder email. And you have to complete your registration first. And once you are done, you will be able to access the courses by creating an account in the learnomicslogic.com, this website. So now Ilya will take over and go through the course materials with you guys. Thank you.
Thank you, Farhana, and thank you everyone who joined today. I will start by just adding to what Farhana mentioned about registration. For those of you that are from NEPI, the National Association of IDeA Principal Investigators, the link for your registration is right here. All you need to do is you need to scroll down and you will find the 4 different programs that are right here, and click, and then you'll have a form that needs to be filled out and somebody will get back to you because we need to confirm with NEPI about membership status and the number of available licenses for free. So with that, let me start my presentation. So one of the things that we're trying to do in these series of training modules is develop asynchronous access to the materials that have been developed and piloted at LSU and LBRN over the past couple of years, and make sure that they can be effective at introducing these important topics and provide a practical step-by-step guide on how to get started with analysis. So I'll briefly discuss the program objectives, how it is structured, where you can find the different resources. We'll also try to make sure that everyone has access to those resources. If you don't, we'll note it down. So if along the way you see me posting some links, those links don't work for you, you don't see what I see on the screen, please put down your name and your email address, preferably in the chat. That way we can get back to you. So the program is going to be structured in several different modules. And so essentially what you will see are live sessions that have been recorded and translated into a step-by-step tutorial. Those are based on sessions that provide an overview of the types of data, the types of analysis methods, and then practical applications, how you can take examples and practice using those example data sets on finding interesting insights that maybe are the same or different from what was already published. And so to achieve that, we essentially designed this process around each one of the topics that is being offered. So for example, this topic is metagenomics. We have 3 elements in the program: the logic, or why are we studying the microbiome? What is the data? What are the interesting questions, research questions that are being asked? What are the applications? What do people do with insights from this kind of data? Then there's the practice step. And the practice step essentially provides us with some step-by-step tutorials. Press on this kind of a button and get this kind of an output, run this kind of code and get this kind of visualization, that kind of thing. And then finally, project. So we'll start by looking at data types and different questions that you can ask. Then we'll talk about analytical approaches. What are they and why are we doing these kinds of things? What are the different pipelines that you have to run to process and prepare the data for analysis? Or what are the actual pieces of code that you can run to get some types of visualization that, for example, are used in publications? And then finally, we'll talk about projects. So how can you find data? What does that data look like? What does the sequencing data look like? What does the metadata look like? And how can we apply everything that we have learned to these examples? So typically, the way we do this is that we do several of these interactive sessions. The sessions are intended to be interactive. That means that if you have any questions, if you think that what I'm saying doesn't make sense, you can put that in the chat. We can stop and we can address those things and make sure that we can all move along. When we have the Q&A sessions, which, as Farhana mentioned, we will have 2 Q&A sessions, we want to make sure that we are helping. So if you have any technical questions or conceptual questions, or even if you're thinking about a research project of your own that you would like to analyzed and apply some of the methods to, please let us know in advance. We would like to know, prepare for that, and be able to share with you some of the things that will actually help. And then finally, we will introduce you to study materials. So our expectation is that people come with different levels of preparedness. Some people come with minimal technical and maybe statistical training, so they are not able to start independently at all. Some people might be more advanced than that. So maybe you have done bioinformatics for a different type of data. Maybe you have analyzed data in general, maybe it wasn't biological data, or maybe you're an experienced microbiologist. So maybe you've read such papers and you kind of know what's expected to happen. You just don't know how to do it independently. So in that case, we are asking that you kind of look at the material as not one size fit all, but find the appropriate place for you. So the study materials are going to be the same pool of materials, but of course you're welcome to choose and pick from them and find something that fits your purpose the best. And then finally, we have these practical assignments. So the practical assignments are going to be of two types. The first type is going to be practical pipelines that will start, and we'll do this today, with demo pipelines. So you don't even have to analyze any data. You just need to understand the logic of each step. How do these steps connect into a series of steps, what we call a pipeline? And then what are the outputs going to look like? How many of them? What types are they? In many cases, these are going to be tables. And to work with tables, we typically want to visualize maybe the results, maybe filter them. And so all of that will be provided as an example of code that kind of tries to illustrate the main things that you should be thinking about when you're analyzing such data. So again, as I mentioned, some of this material is going to be very basic, and that's intended for those that have no background in metagenomics or bioinformatics for that matter. And so if you don't know the terminology, the things that are not maybe clear to you from the very basic things, we have a course called InBytes Molecules, which is an introduction to bioinformatics. How do we describe biological phenomena with data, and what types of analyses are done to that data to make interpretation possible? And the question that we have here in the chat is, can I get the recorded session of this? And yes, after the session is over, we will email everyone the recording of the session. So in this course, Bytes and Molecules, you will find an introduction to metagenomics. So if you don't know what metagenomics is at all, this is a useful place to get started. And just to show you where you can find this. So on the Learn portal, after you have created an account, so I'm assuming that most of you already have an account here. Like Farhana mentioned, it's a free account. So if you do have an account, you go to the Courses tab, and right here in the search bar, you can search for metagenomics, or if you don't know where to start, Bytes and Molecules. So under Bytes and Molecules, right here, Introduction to Metagenomics. The bulk of the material that we're going to be reviewing in the course is called metagenomics. So course number 4, metagenomics. And so what I'll start with today are some of the few lessons in the beginning of this course. But my objective is to help you get started. So you can see what are the types of activities that we're going to be doing. And you can think for yourself where to get started with those materials for your level. And how far do you want to go? So one of the objectives, as Farhana already mentioned, is you can eventually apply some of these methods to your own data or develop a research project around a publicly available data set. I will talk today about some of these data sets, but if you would like to go in that direction, you will need several things. And one of those things is going to be a certain number of points, which, as Farhana mentioned, you can get from completing some of the coursework. Another thing that you will need is access to the bioinfo platform, which today we will make sure that you have that access. And if you don't, by our next session, or maybe by the end of this week, we will make sure that you have that access. And number three, you will need to practice and learn how to analyze the outputs or the tables and visualize them. And so today we'll take a look at some examples, very basic examples, but just to get started, how to load in such data, how to visualize it, and how to interpret it. Okay, so here, as I mentioned, there's an introduction. There are some overview of different types of sequencing data, overview of methods of analysis, and then some coding exercises that we're going to review today together. So the course inside will provide you with a fairly detailed overview of each one of these steps. But today we're going to kind of skip over the details just to get the different pieces in place and to get you started. So this session is not intended to be a full detailed review with practical outcomes. This is supposed to be an orientation, and then you can proceed with whatever kind of piques your interest, or maybe if you want a more structured view, you will see a schedule on the program page. So to get started with metagenomics, I think what we need to start with is we need to understand that the idea behind bioinformatics in many ways is going to be getting started with something practical that takes in some data, provides us with a series of steps, and provides us with an output. And that output is something that we know how to interpret. One of the examples that we'll look at today is an example of a pipeline called the data to pipeline. So this pipeline takes in sequencing data, then looks at what are those sequences in terms of a reference database and how much of each one of those types of bacteria we have, and then visualizes, provides us with a visual output that we can think about, we can try to interpret. As we go into all of these different methods, you need to see that there are many different alternatives to those methods. And what we'll see today is a package that we will briefly take a look at called phyloseq in R that will be useful for different types of visualization techniques that are standard for this one type of metagenomic sequencing data that we will review today. So let's talk about microorganisms and the microbial community. The connection between disease and microorganisms has been established in the 1800s, and already towards the end of the 19th century, laboratory methods for growing microorganisms in the lab culture were developed, helping characterize them with staining. These studies provided important information about bacterial function in isolation. However, it was soon apparent that most microorganisms live in complex communities. Today, we know that the microorganisms, or the microbiota, that make up the human microbiome are not just unicellular organisms living alongside each other, but instead form highly regulated, structurally and functionally organized communities attached to surfaces as biofilms with interspecies collaborations, as well as antagonisms that contribute to ecologic stability. The human microbiota is estimated to be 10 to the 13 to 10 to the 14 of microbial cells. Recent estimates have been making that about 1-to-1 microbial cells to human cells ratio, even though at some point it was reported that there's a much greater number of microorganisms. Some of these figures were updated. And so the numbers are actually derived from the total bacterial cells in the colon, the organ that harbors the densest number of microbes. However, each one of these microorganisms carries important functional properties and genes that contain genomic information. These properties and the genomic information about microorganisms have been of a growing interest to the scientific community, resulting in over 100,000 publications. The majority of these studies have focused on the intestinal microbiome, but as you will see, many different applications of metagenomics have been are available for us to interrogate. So here we can see a recent publication. This is a review. Actually, I'm going to share this link as well. So if you would like to get started, this is a great review recently published. where you can see how different types of areas on our body contain different numbers of microorganisms and what are some of their properties. But also there's a table here that shows the number of PubMed articles on different types of diseases that are associated with the microbiome. So the search was disease and microbiome, and you can see here many different types of disease that microbiome has been extensively studied for. One example is the skin microbiome. So the skin microbiota has the ability to form these highly stable communities. These communities are hidden in kind of areas that are difficult to reach, and so they're not easily removed and changed. And so you can see here some examples we will see some examples of how we can study the composition of skin microbiome that will allow us to determine differences between different regions of the skin or different conditions of the skin, for example, acne and healthy skin. Now, a lot of this information or a lot of such studies are based on our ability to have a baseline. So once we know what the baseline is, what is the healthy condition, for example, or the normal control, then we can start understanding the impact of different types of conditions. For example, specific types of microorganisms that are highly prevalent, or maybe how the whole microbiome changes in diversity, and ask questions about the specifics. And so the ability to establish these healthy baselines is being addressed by large collections of microbial data that are organized in these projects, large-scale projects. For example, the Human Microbiome Project, or the British Gut Project, the American Gut Project. There's the Earth Microbiome Project, right? So all of these different types of projects that seek to collect enough data for us to be able to study and compare to these baselines some of the specific types of experimental data that could be generated. So to be able to analyze such data, we need to have these 4 different things. Number one, we need to know, what does the microbiome represent? And one of the important things about the microbiome is that it doesn't characterize a single organisms, it characterizes a community of microorganisms that have some kind of a relationship to each other. The second thing that we need to understand is, how can we generate digital data that represents this community? And so here we need to speak a little bit about different types of metagenomic sequencing, including amplicon sequencing, whole metagenomic sequencing, metatranscriptomic, and others. So how can we actually study the genomic material in a given sample from a microbiota, a community of microbes, and understand what is happening, or how many different microorganisms do we see? Once we have that sequencing data, we need to understand, what can we do with such data? So how do we actually go from having raw sequences of individual genomic elements to some kind of an interpretable image, or maybe a statistical comparison that we know what to say about? So here we'll talk about diversity, presence and abundance, and maybe different types of functionality that essentially some of these microorganisms can be associated with. And that is essentially one of the objectives of this kind of analysis, is to be able to interpret what is going on. And so that heavily relies on our ability to annotate the different elements that we can find. And here the limiting factor is that there are many more microorganisms than, and a lot of variation within these microorganisms than what it has been annotated. And obviously this annotation continues as an effort into characterization of the different microorganisms. Okay, so again, if there are any questions along the way, feel free to add them in the chat, and those will be very useful for us to continue this discussion. So for metagenomic sequencing, okay, so thank you, Arpita. Where can I find the lecture and Q&A session schedule for the beginner course? So I'm not sure what the beginner course is. For this metagenomic course, we have a page that has all of that information. If you're speaking about the introduction to bioinformatics, maybe you can put your email in the chat and I'll send you that as well. Okay, so today we will take a look at one of these examples, one of the types of sequencing that is commonly used for metagenomic sequencing analysis, or for metagenomic sequencing, is the sequencing of the 16S ribosomal RNA gene. So the 16S, what's called amplicon sequencing, is a technique that is widely used to study microbiome directly in its environment. And it focuses on the 16S ribosomal RNA that can be amplified with PCR amplification. The 16S gene sequences contain hypervariable regions that provide species-specific signature sequences useful for bacterial identification. So the bacterial 16S gene contains 9 hypervariable regions, V1 to V9, ranging from about 30 to 100 base pairs long, that are involved in the secondary structure of the small ribosomal subunit. The degree of conservation varies widely between hypervariable regions, with more conserved regions correlating to higher level taxonomy and less conserved regions to lower levels, such as genus and species. While the entire 16S sequence allows for comparison of all hypervariable regions at approximately 1,500 base pairs long, it can be prohibitively expensive for studies seeking to identify or characterize diverse bacterial communities. These studies commonly utilize the Illumina platform, which produces reads at rates of 50-fold and 12,000-fold less expensive than the 454 pyrosequencing and Sanger sequencing, respectively. While cheaper and allowing for deeper community coverage, Illumina sequencing only produces reads that are 75 to 250 base pairs long, up to 300 base pairs with Illumina MySeq, and has no established protocol for reliably assembling the full gene in community samples. Full hypervariable regions can be assembled from a single Illumina run, making them ideal targets for the platform. So here you can see that the 16S ribosomal RNA, about 1,500 base pairs long, and then we have these V1 to V9 conserved regions. And so choosing some of them, one or a couple, makes it a good candidate for these kinds of studies. Now, to analyze such data, once such data is prepared, we can reference the different databases that contain sequences of the 16S ribosomal gene, like you can see right here. Green Genes, the 16S ribosomal RNA gene database and tools, or the SILVA, a comprehensive online resource for quality checked and aligned ribosomal RNA sequencing data. So essentially what we can do is, once we can generate sequences of those 16S ribosomal RNA genes or the hypervariable regions of those genes, we can search these databases and find the best matches to then provide annotation of what have we actually found. So the 16S taxonomic classification is based on the ability to sequence a particular targeted region, which is amplified. So we actually lose the actual specific abundance of that particular gene that is present in the sample. And we can now look at composition, so the proportionate abundance. And so once we look at that annotation, we can use the different levels of taxonomic classification to then assign proportions. And when we talk about baseline studies, we can look at individual regions on the body, and we can, for example, establish some composition that is stable across several individuals. So this is just one of those sequencing techniques. In fact, there are several different other approaches. For example, you can look at RNA-based approaches, metatranscriptomics, or there's an ability to sequence all of the, not just the 16S, but try to sequence, especially using long read sequences, sequencers that have become more accessible and cheaper in the recent times. We can actually sequence either the full 16S ribosomal RNA gene, which could provide us with ability to better characterize the lower level of taxonomy, so it's more detailed, or in some cases even be able to assemble the actual full genome, which could be extremely interesting because we can only rely so much on knowing one individual gene only and assuming that everything else stays the same. Now, another kind of ability to study this kind of data is to try and see not only which genomes are present, but also which genes are being transcribed. And so with metatranscriptomics, we're relying on RNA. Here, it's important to separate the genes that are being produced by the host as opposed to the actual microorganisms that are present. And then there's also mass spectrometry, where we can see analysis of the proteins and the metabolites that are being produced. Okay, and Raphael is asking whether 30x coverage is enough. So again, many different applications, but if you have a specific question, enough for what, we can probably talk about that as well. So ultimately, though, when we have this idea of what is present and maybe in what proportions, ultimately, I think the whole field is trying to move in this direction of not only what does that mean that we have more of this microorganism or less of that microorganism, but what does it actually mean functionally, right? How can we use this understanding of composition or what genes are being expressed to ultimately say, okay, what does this mean? What is that functional component of this type of information? And for that, I think the whole field is moving into greater detail. And that means combining more and more different types of data to be able to ask these much more specific questions and apply them to things like described here, the impact on the immune system, or maybe interaction with drug metabolism and other questions like that. Okay, so let's do a little overview of where we can find such data and what does this data look like, or what can it be used for. So again, not a long time, because we will spend a little bit more time on some of the practical things so you can try and get started, but we'll do a brief overview. So one of the things that you will find very useful, I think, and intuitive is this American Gut project. The American Gut Project is a citizen science project dedicated to metagenomic sequencing of world citizens that are contributing their microbiome samples to the study of healthy or almost healthy microbiome variability. So the idea is, can we ask people to collect their microbiome samples, skin and gut samples, send them, sequence them, and then we are starting to collect this natural variability. And one of the examples that you will find in the American Gut Microbiome Project, which we will actually try and do in this program, is look at an individual single sample, right? So let's say I contribute my sample. How can I look at my sample? For example, you can see right here, my sample is this first bar compared to the average similar diet. So people that are on a similar diet like myself, same gender, similar BMI, age, and some individual, Michael Pollan in this case, so some individual that we can compare. And as you can see, the comparison consists of different colors here that are annotated right here. And for kind of understanding my individual sample, now we can start looking at the most abundant microbes at different levels of taxonomy. So these different levels of taxonomy now can give us some reference that we can try and see what is known about these individual microorganisms or taxa of microorganisms. Down below, you'll see how different sites, so HMP here stands for Human Microbiome Project. So that's where we get the reference, kind of how does this compare to the population? So how can we compare to some of these other sources of information from different body sites, different ages and populations, and the American gut population. So this is a project that I think is fascinating, provides us an insight into what could be done just kind of to understand what's going on. But also, together with this information, you can find metadata. So that was sequencing data. What are the microorganisms that are present? This is an example of metadata. So this is metadata from the Human Microbiome Project. The Human Microbiome Project contains a lot of different variables that are being collected from individuals. And in this one example, we are using a comparison between people that exercise rarely and exercise regularly. So here you can see we can control for certain conditions. For example, they don't have the liver disease. They are considered to be a healthy subset of people. They rarely or maybe regularly supplement with vitamin D. But the biggest difference here is that the exercise is never versus daily, right? So one of the questions that you could ask from such data is, if we control for as much as we know for the different factors, could we find some individuals that are different in one particular way, and then based on that difference, see whether there is or is not a difference in their microbiome. So just an exploratory, simple question. And we'll take a look today at some of the ways that you can start thinking about these questions using this kind of data. Another example that we will look at is how the skin microbiome varies between acne and what's called perioral dermatitis. So a very common condition that a lot of people have either experienced or might continue to experience. The perioral dermatitis is more of a rash, and acne is a more acute condition. And then, of course, we also have areas of healthy skin. So one of the questions could be, how do these microbes essentially interact with the skin itself in inflammatory acne, right? So how do they cause inflammation? And so here in this discussion, we can also download the data, and we'll look today at where this data could be found and try to understand what are some of the differences. So in this case, the difference is represented in this image right here, which is a non-metric dimensionality scaling plot that allows us to see different samples positioned in this two-dimensional space and maybe show us specific grouping based on this annotation, kind of giving us a sense overall whether these samples are different or not. And here we have 3 different samples: adult acne, none, which is healthy control, and perioral dermatitis. Another more complicated study in terms of how it was performed and exactly what was done to develop the data, to kind of prepare the data, is a study that was published in 2015 that spoke about obese gut microbiota induced neurobehavioral changes in the absence of obesity, right? So in this case, the researchers were trying to separate the obesity itself, for example, resulting in some genetic predisposition or maybe in some kind of a diet, from the impact of the actual microbiome or the the composition of microbiota. So could we actually take the microbiota of mice that were fed a high-fat diet, use that microbiota to implant a specific composition into a healthy mouse, and then see whether that led to some neurobehavioral changes? And if you read this publication, you will find that, yes, they found an association with anxiety and these kinds of patterns of behavior. But what we can try and do is we can try and take this data and try to understand differences between several different types of data that are available with this publication. Now, of course, these kinds of publications illustrate some of the approaches. They can become more and more complex in nature, and you will find that there is a great interest in trying to understand the functionality, the functional applications, and even develop treatment. For example, here you can see a treatment for Alzheimer's disease that targets the microbiome. So different types of applications of microbiome, obviously going beyond some of the basic research questions and trying to incorporate the microbial signatures into practical applications, including clinical applications like treatment or even understanding of the impact on other types of treatments, like, for example, immunotherapy. Today, living in the pandemic, of course, we all might have questions about, well, what is the relationship between the microbiome and COVID-19? And here's an example of a study where the microbial diversity is being studied for long COVID and for susceptibility. So here you will find high-risk groups succumbing to COVID-19. As a result of the pandemic providing us with such tremendous quantity of data, some researchers decided to collect that data as well, and found some differences between the length to recovery and speed of recovery as well. The microbiome is also used not only in medicine, but also biotechnology and agriculture. For example, microbial fertilizers, MFs, constitute an emerging class of organic fertilizers and consist of dried microbial biomass, for instance, produced on effluents from the food and beverage industry. They have been shown to provide unique benefits, especially in harsh conditions, at the same time with minimal environmental impacts. Metagenomic studies on agricultural applications of microbiome are showing promise, and many have already started using this approach to improve yield and resilience of crops. And so the types of applications of metagenomic sequencing and sequence analysis really continue and go on. And what we'll see is we will see how these types of metagenomic studies focused on the microbiome as a whole are very different from the way microorganisms can be studied in isolation. But just to compare and contrast, here are some examples of microorganism studies in isolation, not as a community, where you can see drug resistance, viral adaptation, or association with a particular phenotype that could be studied once you have the full genome of the microorganism. Okay, so now let's talk a little bit about some of the practical things that we can do to analyze such data. And again, this is not going to be a very detailed overview, but it should provide you with a high-level understanding of some of the practical activities that you should be able to do after completing this training, and how you can continue thinking about some of the research questions that we covered up until now, and try them on different types of data sets. So the first thing that we're going to do is we're going to try and learn this data to pipeline just by understanding the sequence of steps. How do we go from raw data to process data? And then what are some of the outputs? What are some of the visualizations that we can get at the end? So to do that, let's go to the server, which is server.tbio.info. This is a platform that has a lot of the methods implemented in a user-friendly environment. And in addition to that, you will get some demo examples where you don't even need to run any analysis. You just need to understand and visualize some of the data. So please log into this platform. If you don't have a login, for example, you just joined today, you haven't really registered yet, You won't be able to do this together with us now, but if you have created an account on Learn, within 24 hours, you should be receiving your account details. So just to get a sense of how many of you already have an account, could you put number 1 or yes or something in the chat so I know? Okay, great. So 2 people. So if you do not have an account, but you would like to have one for continuing, please email support@pine.bio. Okay. Okay, so unfortunately, those of you that don't have an access, you won't be able to do this practically, but you will see that these are very simple examples. So once you get the access, it will be easy for you to replicate. So what we'll do here, and we've changed some things around, you will notice that some things have changed not so long ago. We have moved the metagenomics section right here. And what we will do today is we will just take a look at this last example called Amplicon 16S, 18S. So if you click on this dropdown, you will open this section right here. And this is called the data to pipeline. So you will find right here in the middle a section called data to. Now, we're not going to go into a lot of detail about this. What I want you to see today is just how to run a demo pipeline, what are the inputs and outputs, and what does it mean? Where do you go from there? So here you want to click on Start. You can see what we covered today, kind of what is being sequenced. The data to pipeline is specifically designed for pair-end metagenomic sequencing data for Amplicon 16S ribosomal RNA. So here, the 16S ribosomal RNA hypervariable regions are being sequenced, not the full gene. And then one of the objectives of this pipeline is to actually denoise the actual sequences. So the more precise the sequences that we have, the better we will be able to match them accurately to the reference database. And then if we have an accurate match, we can then also analyze differences in composition, or maybe how they are related to each other phylogenetically. So here, an important step is to understand the challenge, right? And one of the challenges is with this great diversity of microorganisms that we are studying, that we are sequencing, there is expected to be a lot of variation. And because of that variation, it's important for us to differentiate between technical variation, like noise in data coming as a result of inaccurate sequencing, maybe because we don't have a good coverage, or maybe because there is a lot of diversity. How do we actually understand the biological diversity that we have? We have to denoise and kind of eliminate the mistakes. And to do that, we need to merge the pair-end reads and then combine them into what's called operational taxonomic units. That allows us to eliminate a lot of the variability that is inconsistent. Unfortunately, it also reduces a lot of the reads that we will have, but we are only going to be keeping reads that are accurate and could be matched to something. So once we do this, I'm going to quickly go through some of these steps. We want to assign those to the right reference in this database. And assigning them means that now I can not just speak about individual OTUs, operational taxonomic units, but I can group them by whatever level of taxonomy I was able to annotate them by. And one of the challenges is going to be the lower we want to go on the taxonomy scale, the less information we might be able to get because of these hypervariable regions and their variation, but also because of the coverage and how precise we know which one came from there. Now, from this point on, we essentially have several different tables. Those tables include a table of OTU abundance, or how much of a particular operational taxonomic unit I have proportionately in each sample, and the annotation of where does that OTU map to from the reference database. Because I have the reference database, I also know the relationship between them. How close are they to each other? Could they be grouped in some way? How far away are they evolutionary from each other? And then I can perform downstream analysis, which includes building that phylogenetic relationship with ape, and then visualizing the results with phyloseq, which is a package in R that allows us to make visualizations and all kinds of statistical analysis that then we can interpret in terms of differences between groups of samples or looking at individual samples. So once we do that and we click on end right here, we will look at some of the output files. So once you click on OK, right here you will have a folder called download pipeline output files. Okay, how many of you were able to get to this last screen that I have? Okay, perfect. Rafael, did anyone else? Okay, if you did not, again, I'll just repeat the steps really quickly. So from areas of analysis, down here, amplicon 16S, 18S, you click on this drop down, go into the demo, start, QC filtering, replication and count, pair merge, cluster consensus OTU, remove chimera, assign taxonomy, ape, and then phyloseq. So after this, you click on end, and you get the download pipeline output files. Open this up, and now you have several different things. And these are the kinds of results that you're going to be getting from a pipeline. These results will include tables, TXT files, for example, plots, like these PDF plots right here, FASTA files of actual sequences, and those are the major types of data that you will have to deal with. Okay, so as an example, right, this is going to be explained in much greater detail in the course. So when you go into the metagenomics course and you want to understand what was it that we were doing, right here you have a practical 16S metagenomic analysis lesson that you can review, understand each one of the steps, and see all of the results in one place. But before you jump in there, you want to go through all of these different sections to understand what kinds of sequencing data do we have? What are the specific ways to analyze them? And then even practice some of the basics so that you understand what is going to be in an individual sample file. And to do that, let us take a look at another example. So let's go. Now, hopefully everybody has a good sense of what we can do on the platform, just what kinds of things we're going to be doing. And here we're going to be doing something else. Here we're going to try and run a piece of code in R. And at the end of this session, you can download this code and run it on your system as well. So what I will do is I will go into this lesson 6. Okay, this is the link. And we'll just go through this lesson with a little exercise at the end. Okay, so please let me know once you're on this link. We're going to take just a few minutes. It's not going to be a long time. All right, perfect. So here we have a different problem. There we had multiple samples. And the question was, how do these groups of samples compare to each other? In this case, we will just take one sample. So we don't have anything to compare to. We just want to understand what's in it. And to do that, we will use a different pipeline. So this is a pipeline that is going to do something very different. Here we will take full 16S ribosomal RNA gene that is sequenced. We're going to align it to a reference database of all 16S ribosomal RNA genes. We're going to count how many do we have of what kind, and we're going to annotate them. Okay, and so here, the basic table that we're going to be dealing with looks like this. you have individual elements, genomic elements, for example, 16S ribosomal RNA. And you can see how the kingdom, phyla, etc., is organized according to the different taxonomic classification with how many of each kind do I have. And then you will also be able to annotate, right? So separate them and see how many do you have of different types of, for example, phyla, right? So if we just focus on phyla, we can see how many of each kind do we have. Now, they will come up many times, right? Because multiple organisms fall under different phyla. So we need to combine them together. So after we combine them together, we can calculate percentages. What's the percentage of a particular type from the total that we have? And obviously, we can visualize such data, right? We can prepare some kind of a visual representation, and that is sometimes more informative. We can do a pie chart, or we can do a bar plot. So essentially, we can see what is happening with our data. And so this lesson is to be able to do these simple steps in R. If you already know R, you know that for many different applications in R, there's typically a library that already contains many of the functions that you would like to use. So you don't actually have to reinvent the wheel. Some of these packages are very specific, like phyloseq. They just produce a set number of plots that are useful for a particular application, like, for example, phylogenetic analysis for phyloseq. But some of them are more general libraries, and those libraries include ways to visualize data and create different plots, or maybe calculate different statistics for different types of things that you can find. So here we'll do something very simple. We'll learn how to load our data, prepare a pie chart, and then we'll make a bar plot, because bar plot could actually be more easy to interpret visually than a pie chart. So the objective would be just to understand how many of each kind do I have in a particular single sample without having to compare to anything. So here we have some code, and let's just go through this code really quickly. We load a library. This library is to load an Excel file. Now, sometimes you will have CSV files, TXT files, Excel files. So you need to be able to load those data and load them into your environment, into your script. Here, we'll do it right here on our server, but you can also do it in R. So then we want to sort the data. And sorting the data is useful because instead of looking at images that look like this, we can kind of organize them to all look like this, from highest to lowest. And once we do that, we then create percentages, make a pie chart, and also make a bar plot at the end. So let's see how difficult is it for us to perform all of these steps, including adding some color here as well. Okay, so here you have a console, which is where you run the code. And in this console, you already have all of the data and all of the script loaded in. In fact, if you want to use this data set on your computer, you might want to download it right here. So just right click on this link and download linked file. If you want to see what happens when you run this code should run in exactly the same way on your computer as it does here. The only thing that you need to remember is that we're using a library. Yes, you can run it on RStudio. But in RStudio, you will need to have, the only library that we're using is this additional library, Excel, to read Excel files. Okay, so let's try to run it. And you can see that, first of all, we have a success, right? We correctly have the answer. And right here, we have the results. So this is a pie chart that we created, and this is a bar plot that we created. And this bar plot is already annotated. Now, let's try how to change just one little thing so that we know that this is actual code that runs and not just an automatic button. So one of the things that you can see here is that when we create a pie chart or a bar plot, we actually give them a title. Main is a title. And here we also make different dimensions for our plots. So we're going to do two things. We're going to increase the dimensions because this is kind of small. And we're actually going to change the title. Okay. So here we're going to change this to pie chart. And here we're going to add bar plot. And you can try to do this in RStudio as well. Okay, so here you can see that we also add the label to our x-axis. And so when we look at the x-axis right here in the bar plot, you can see bacteria type. Okay, but actually the type is by phylum, right? So here we want to add phylum. Okay, now once we do this, oh yeah, we also wanted to increase the size. So let's do it like this. And let's run it again. Okay, so here, pie chart, right? We changed that. And here, we also changed phylum and bar plot right here. And I noticed, by the way, that I capitalized the C so we can make it smaller. So this is an actual piece of code. If I want to take it to my computer, what do I need to do? Copy, right here is the copy code button. Open RStudio. If anyone doesn't have RStudio, we have a little tutorial on how to install RStudio. And then you can run the exact same code. Obviously, you would need to set up your environment to have the library and to have the file. Okay, so here's our RStudio. Let's make it bigger. Create a new script. Paste it here. Okay, and here, right, we have this library. I already have it installed. Let's run it. Okay, object labels is not found. So right here we have, so I think, what is happening? Let's run it like this. Okay. Like this. And run it like this. All right, and so there we have it. It also works in RStudio. Okay, so to summarize, because we're already going over time, let's wrap this up and summarize what we have done today. The objective of this program is to provide us with methods of analysis that we know how to perform and we know how to interpret. Today, we ran a demo pipeline called the data-to pipeline, and we saw this data-to pipeline in terms of the inputs and the outputs, kind of understanding the logic of analysis. Now, there are many other types of analysis that we can do, and we looked at this annotation of 16S ribosomal RNA. We have the kind2 pipeline as well, and we have this data-to pipeline. So we can look at those different examples and how they approach the same problem with different other methods. We also looked at this simple example of visualization. How can we visualize the data that we have from sequencing? And we saw that we have this abundance of proteobacteria in this particular sample. Now, what do you do moving forward? So I recommend several different publications that you can go through to kind of get started moving further into this field of metagenomics. There's a good review of the Human Microbiome Project, which contains a lot of 16S ribosomal RNA data used in a variety of studies. You will also find here a paper, I'll share the links in just a moment, bioinformatics for microbiome research, concepts, strategies, and advances, and also explore the actual human microbiome project as well. So this is a collection of data that has 16S as well as metagenomics as well. And you can see different types of collections within this project. So let me get you these links real quick. Okay, so here are those links. Okay, now moving forward inside the actual tutorials, you will find these different additional resources: an overview of sequencing, methods of analysis, different project examples, including some exercises that you can do. And then hopefully after you complete that kind of training, you will be able to apply this to a data set of your own. So we do have a whole project where you can go through several lessons on how to develop an independent research project, including where to find data, how to plan for your analysis, and if you need help, how to submit it to some of our mentors to give you advice and direction on how to develop an independent research project with omics data and bioinformatics. That's all for today. Excellent, Rafael. I'm glad to see that you were able to run this in R as well. I will be available for questions during the Q&A session. Yes, you will learn how to do alpha diversity and beta diversity as well. We have that in the lessons, and you will see it in PhyloSeq as well. So please go through the lessons. Again, this was not a lot of information just for us to get started, but as you come up with any questions or challenges, technically or otherwise, please email us. As you can, if you are at LSU, please reach out to Farhana. If you are not from LSU, please reach out to support@PineBio, or you can reach me directly, also Ilya@PineBio as well. All right, excellent. Thank you, Rafael. Glad that you were able to join. And this recording will be shared with all of you as well. So we will email to everyone who has registered for the course, and we hope to see you again for the Q&A session. Thank you, everyone. Take care. Thank you.